There's a very nice paper that's just been published in PNAS using some structural biology techniques to look at filoviruses. Now, filoviruses are called filoviruses because that means thread-like, and as you'll see, these are very long, thin viruses. So the paper has actually looked at two viruses, Ebola virus, which is a very highly pathogenic virus, and a closely related virus known as Marburg virus. And they've used two techniques to do this. They've used cryo-electron microscopy, where virus particles are frozen. So this is a very gentle technique which doesn't disrupt the structure of the virus. And then they've used tomography, which is using computers to reconstruct images and build three-dimensional models of the data. And using these techniques, what the researchers have been able to do is examine these long filamentous virus particles you can see here, and they find some interesting things. They find that the diameter of Ebola virus and Marburg virus is slightly different. Ebola virus is slightly smaller in diameter than Marburg virus, and you can see that here in these two graphs. But the interesting thing is when you look at the length of the virus particles, there are two populations. These relatively shorter Ebola virus particles, although these are still long particles, and these much longer ones. And they're able to show that this difference is due, the difference in the diameter of the two viruses is due to the nucleocapsid protein, the protein associated with the RNA virus genome in the center of the virus particle, while the difference in the lengths of the virus particles is due to the RNA genome of the virus. So we have two genome classes here separating these two populations. And putting all of this information together, what the researchers have done is to build an assembly model for filoviruses, in particular Ebola viruses. So this is a very nice use of structural biology to probe the structure of these viruses, which we really don't know enough about at the present time.